In this video, we will discuss about the pathway contribute to the MMP9 expression. So, when the tumor progresses, there are many pathways that gets upregulated, and one of that common pathways that are found to be upregulated in the tumors, but not in the not that much in the healthy cells, or is the MAPK pathways. So they wanted to check whether this MAPK pathways has some kind of contribution to this MMP9 expression or not. In order to check this hypothesis, what they did is they first make a cell line in this NPC039 and NPC076 that has this luciferase MMP9, so that they will have the MMP9 data expressed on those cells will now have this luciferase signals, which makes them easy to quantify the impact of these pathways on the MMP9 using luciferase signals. So the genes that are involved in these MAPK pathways are being targeted using the individual inhibitors. For example, this U is the inhibitor for ERK12 pathways, SP is the inhibitor for p38 pathway, uh, not and this SP, yes, SP is the inhibitor for JNK pathways, and this QN is the inhibitor for NR-kappa beta pathway. And they used in the panel A the gelatin zymography to analyze the impact of these inhibitors on this MMP9, and MMP2 is used as a control. So they are able to see that in almost all the inhibitors here, although it looks like high compared to the other inhibitors, but compared to the control, that is with PMA treatment alone, this signal is low. This is quantifications is shown in the panel B. So let us first go through the panel A. So for both this NPC039 and NPC036, when they use the PMA, and before that they use the cells individually treated with the inhibitors, 4 different inhibitors, and they are able to see that compared to the control, that is compared to the PMA treatment alone, the amount of MMP9 goes down for both the cell lines. And they did the similar studies. Instead of using PMA, they used macrophages condition media and all the inhibitors. So in that case also they are able to see the same signals, that compared to the control, the individual inhibitors of this MAPK pathways shows reductions in the MMP9. And these things they have quantified, and it is evident that the decrease is statistically significant compared to the case where they used only PMA. So this indicates that these genes that are involved in these pathways has a role towards this MMP9 expressions. After that, they checked the impact on this intact N-cadherin or full length N-cadherin and the cleaved N-cadherin part, that is CTF1. So when they check the intact N-cadherin, they see increase in they see almost the similar levels of N-cadherin, although there is a slight increase, but that increase is not statistically significant. And similarly, they also checked it for this cleaved N-cadherin region, that is CTF1, for the inhibitory conditions, and they can see that this cleaved N-cadherin or CTF1 signals are low when they use the different inhibitors. That can be seen here. And in that case, they show the results for the 3 inhibitors only, whereas in the panel A they did the experiment with 4 different inhibitors. So anyways, the result shows that is consistent. The results are consistent with this results that are shown in this panel A and panel B, that the genes that are involved in this MMPK pathways has a role towards this MMP9 expression, as with by inhibitions of this any one of these genes will result in the decrease in the N-cadherin cleavage and simultaneously increase in the full length N-cadherin signals, although this increase and decrease is not statistically significant, but we can see the trend. And however, the results in the panel B, which corresponds to the results for the panel A, are statistically significant. That is, MMP9 productions goes down when any one of the genes that are involved in this MAPK pathway, not anyone, these 4 genes that are involved in this MAPK pathways are being targeted. We can see the decrease in this MMP9 signals. So this is how they come to a point or come to a conclusions or come to a logical understanding that this MAPK 9 pathways has a contribution towards this MMP9 expression. So if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.